So let's talk about biomolecules. We're talking about carbohydrates, proteins, and lipids. This is what the syllabus objective says. We need to talk about the structure and function. And we're also going to touch on the enzymes that are involved in chemical digestion. First of all, with carbohydrates, they're made up of carbon, hydrogen, and oxygen atoms. And it's in this ratio here. So for every carbon, we've got H2O, so water. So the name carbohydrate comes from hydrated carbon. So it's carbon and water. The building block of carbohydrates is called a monosaccharide. So this word monomer, you're going to see for each one of these macromolecules. The monomer is the single building block. So in carbohydrates, that single building block is called a monosaccharide. Mono being singular, saccharide being sugar. So we're looking at glucose and fructose as examples. Put two of those together, we get a disaccharide. So for example, sucrose. Sucrose is like table sugar. And then we've got these things called polysaccharides, which are poly meaning many. So there are many of these sugar molecules joined together. And we've essentially got three key ones. We've got starch, which is the way that carbohydrate is stored in plants. So thinking about potatoes and carrots. So that's starch. Lots of those sugar molecules joined together. Glycogen, this is an example of glycogen here. Animals store carbohydrate in the form of glycogen, and that's highly branched like this, so you can pack a lot of it into a small area. And then we've got cellulose, which is actually a structural carbohydrate in plants. It forms a large part of the cell wall. So the function of carbohydrates is the primary source of energy. Glucose is used as the source of energy in cellular respiration. Glycogen is how energy is stored in muscles and the liver. And energy storage in the plants is starch. And as I said, cellulose is a structural element. And cellulose is a structural support in plants. Now with proteins, the building block or the monomer is the amino acid. And we've got 20 different amino acids that are involved in proteins. And what differs between them is this R group. So this is an example of an amino acid here. We've got the amino group here. We've got the carboxyl group. But here in the middle, we've got this thing called the R group. And that varies with each of the different amino acids. And it's the side group that determines how the amino acid folds and bonds together with other amino acids for proteins to have their function. So there's actually four different levels of structure of proteins. The primary structure is the sequence of amino acids, which is dictated by the genetic code. The order of the amino acids is dictated by the genetic code and is put together in protein synthesis. And each of the amino acids are joined by a peptide bond. And we call that primary structure a polypeptide chain. The secondary structure is when that polypeptide chain either folds or coils into a shape and it's held together by hydrogen bonds between the amino acids. Many of our structural proteins only have a primary and a secondary structure. They don't have a tertiary structure. The tertiary structure is when a protein has a 3D shape. And it's like we also call them globular proteins because they fold into like a ball. And these are our functional proteins like enzymes, etc. Now, the important thing about these R groups, they've all got different R groups and they're all capable of making different types of bonds. And it's those that dictate the type of structure or the shape of the final protein. So you can see here we've got lots of different types of bonds, hydrogen bonds, we've got disulfide bonds, ionic bonds, etc. And so it's the order of the amino acids dictates which side group goes where and how it folds and interacts to hold into this 3D shape. Now, if we have mutations in the genetic code, it means that we might be forming different amino acids which have different R groups, and it might mean that there's a different shape to the protein, and hence it might affect the function of the protein. So what is the function of proteins then? We have structural proteins, which are long fibrous proteins that provide strength and support, like collagen, silk, keratin, that sort of thing. They generally stop at this point of having a secondary structure. But then we've got the globular proteins, the ones that are folded into a globular shape, and they are functional proteins. So, like, for example, enzymes, hemoglobin, insulin, that sort of thing. Now we have lipids. There's 3 different types of lipids. Triglycerides, they have glycerol plus 3 fatty acids. So this is the triglyceride here. And this is the classic version of a lipid that we would consider as fat. Then we've got phospholipids, which of course form the cell membrane, the phospholipid bilayer. And we've also got, and so they've got the phosphate head and the 2 lipid tails. And then we've got steroids, which are, an example of that is cholesterol, which is also part of the phospholipid bilayer. So the function of lipids, they are long-term energy storage, but that also provides insulation, protects organs, and also keeps the body warm. We also see it, of course, in the plasma membrane, and many hormones are lipids as well. So that's our 3 macromolecules. So organisms produce those macromolecules, proteins, etc., from the monomers, and we get the monomers from food. So we eat protein and break that down into amino acids, and then the cells then put those amino acids back together as proteins. We do the same thing with starch, etc. We break it down into glucose, really small molecules, allowing it to be absorbed through the small intestine into the blood. Now, so we have mechanical digestion and chemical digestion, and the chemical digestion largely occurs due to enzymes. And so these are catabolic enzymes because they break down. We have 3 enzymes here. Remember, enzymes end in ASE, and often their name gives us a bit of an idea of what they do. So lipase breaks down lipids into fatty acids and glycerol. And that happens with the saliva in the mouth and also in the small intestine. Amylase breaks down starch into sugars, and that occurs in the saliva and the small intestine again. And protease, which breaks down proteins into amino acids, and that occurs in the stomach and the small intestine.